Good afternoon, everyone. I am Dr. L.B. Lahirupibode, clinical geneticist, senior lecturer, Faculty of Medicine, University of Laguna, and the Secretary of Human Genetic Society in Sri Lanka. I welcome all of you to the monthly academic webinar of Human Genetic Society. With the continuation of the vision of the Human Genetic Society to promote genetics and genomic education, we are organizing a series of monthly academic webinars as a part of our web-based e-learning platform to keep everyone updated on current topics. Today, the topic of the webinar is the advances in pre-implantation genetic testing. Today's speaker is Dr. Fiona Olvitta D'Souza. Fiona Olvitta D'Souza is a clinical embryologist and head scientific operations, clinical genetics, Anderson's Diagnostics and Labs, Chennai. She was a research assistant and a senior research fellow, Department of Clinical Embryology, Kasturba Medical College, Manipal University, Manipal. She is a life member of Manipal Association of Clinical Embryologists and Indian Society of Assisted Reproduction. She has received many awards and grants and published several peer-reviewed publications on pre-implantation genetic diagnosis and assisted reproductive technologies. Her areas of specialization are clinical embryology and assisted reproductive technology, chromosomal aberrations in male infertility and pre-implantation embryos, pre-implantation genetic testing for aneuploidies, structural rearrangements and mutations, pre-implantation embryo metabolism, non-invasive embryo quality assessment, and DNA damage response in pre-implantation embryos. So I warmly welcome Dr. Fiona to start her presentation. Over to you, Fiona.
Thank you, sir. Firstly, I would like to thank Dr. Hiro for the introduction and this opportunity to address you all on this forum. I would be mainly discussing on the basics of pre-implantation genetic testing and how we go about this testing and some recent advances in this technology. To begin with, all of these genetic testing in pre-implantation embryos started from over 4 decades of assisted reproduction, where we struggled to enhance live birth rates and pregnancy rates. There were several modalities that were changed and improved over these 4.5 decades, and we shifted to different forms of embryo selection and still were able to achieve a live birth rate of only about 36 to 40%. These limitations led to a strong drive in finding out alternate ways to select embryos for transfer in IVF cycles. There was a systematic review published way back in 2011 that showed that 73% of all human embryos resulting from IVF contain some or the other aneuploid cells. And also, it is a well-known fact in embryology that morphologically normal-looking embryos, they are known to carry chromosome anomalies. So looking at them, it is really not possible to ascertain the genetic status of these embryos. For this purpose, we went on mainly to test the genetic content of pre-implantation embryos. To select these embryos, initially way back in the 1990s, we started with fish for a targeted of about 5 chromosomes. This was what was known as PGT 1.0. That is the initial part of it, where day 3 embryos were biopsied after IVF, and they were used for detection of aneuploidy of only 5 chromosomes by fish, and then they were transferred. But this does not, they did not give any good amount of improvement in the quality of IVF success. However, as time went by, new technologies came up with array-based CGH, QFPCR for comprehensive chromosome screening, and in early 2000, next generation sequencing came into the picture. So right now, next generation sequencing is the most widely used platform for genetic testing in embryos. And it also came right in time when IVF related technologies were completely optimized to culture the embryos for 5 days. Vitrification of embryos was very well established, and the skill set of embryologists was honed to do efficient trophectoderm biopsies. Now, all this was established as the new version of PGT, that is PGT 2.0, where trophectoderm biopsy was performed and genetic testing was performed for all chromosomes as opposed to the limited chromosomes when it came to fish. So as these things progress, just a quick word about what's on the horizon also. There are advancements in genetic testing now that are leading us to helping us to testing for the mitochondrial DNA. There are advanced workflows that help us address mosaicism. We are also looking at epigenetics because long-term culture of embryos for 5 days in the IVF lab has been raised, causing some raised concerns on epigenetic modulations. There are studies being conducted right now to address transcriptome of the embryo. And of course, the most recent advancement that is almost on the verge of entering the market is the non-invasive assessment of embryo genetic material from the culture medium. So what are the indications for genetic testing in embryos? This is a very enhanced and next level of testing, which is not given to all patients, mainly because of the financial burden, and we don't really need to do it when it is not indicated. So the indications for pre-implantation genetic testing mainly are addressed to uh, improve patients with recurrent implantation failure, advanced maternal age, if they have a previous history of genetic anomalies in aborted fetus or any aborted fetus that has shown a genetic anomaly, a family history of structural chromosomal abnormalities on the karyotype, or male factor infertility, because if poor quality male factor in the sperm, as if we end up with cases like oligosteroteratospermia or non-obstructive azoospermia, it is known to be associated with increased level of sperm aneuploidy, and even a 2 to 4% increase in sperm aneuploidy can cause about 60% increase in embryonic aneuploidy. So that is why that is important. Another most important emphasis that we put on is female age. So this was a very beautiful paper. It was a review of about 15,000 biopsies that clearly showed how aneuploidy changes with female age. So even when you have patients in the 30 to 33, 35-year demographic, we're looking at close to 30 to 40% aneuploid embryos. And this always increases with time. So it's not just the IVF conditions, it is also female age that significantly puts embryos at risk. And if you look at patients in the 40 to 45-year-old demographic, you have 65 to 85% embryos that are turning to be out to be aneuploid. And these embryos don't implant or undergo miscarriage. Again, it's a lot of trauma to the patient to go through all of this. Therefore, PGT-A was mainly designed to improve implantation and pregnancy rates in advanced maternal age and patients who are suffering from recurrent IVF failures. It was also showed by a number of papers that PGT-A significantly reduces the time to pregnancy and the time to live birth in women, even if there is diminished ovarian reserve. So there was a study that compared IVF with PGS and IVF without PGS. There are plenty of studies showing similar kind of data. So as the age improves, obviously IVF pregnancy rate is dropping. That you can see by the blue bars. But what is interesting to see is we are able to provide a consistent and excellent pregnancy rate when PGS is done in coupling with IVF, especially for patients with advanced maternal age. Coming to the nomenclature, there's a lot of confusion regarding what to call it now, because conventionally embryo testing, pre-implantation embryo testing was known as PGS and PGD, and now there are so many different types. And just to clarify on what these types are, to begin with, the new nomenclature addresses all genetic testing in embryos as PGT, that is pre-implantation genetic testing. And the next abbreviation after that specifically mentions what exactly we are looking for in these tests. So if it is PGT-A, that means I am looking at chromosomal aneuploidies, if there is increase or reduction in the number and content of chromosomes. PGT-SR is addressed to structural rearrangements. This is when we are looking at translocations in the embryos. PGT-M is going to address monogenic or single gene disorders where we have a point mutation and we're very clear on what we are looking for. PGT-M is always done along with PGT-A. PGT with HLA is for HLA matching when parents are looking for a donor child for an already affected sibling. And this is encompasses both PGT-A, PGT-M, and then you do PGT-HLA. New techniques on the horizon that a lot of people are hearing about is niPGT, that is non-invasive PGT, which uses the culture medium for genetic testing. We do not have to perform any invasive biopsy as opposed to the previous 4 types of PGT. Minimally invasive PGT is when you extract blastocele fluid. So the blastocyst is a 3-dimensional structure with trophectoderm on the outside and inner cell mass on the inside. The inner cell mass gives rise to the embryo proper, and there's a fluid-filled cavity at the center of the blastocyst, which contains blastocele fluid, which is actually secretions from all the surrounding cells of both the ICM and the trophectoderm. Another recent technology that is on the horizon is PGT-P, that is PGT for polygenic disorders, and this is also labeled as ePGT, that is expanded PGT. So these are one and the same thing. So how do we identify patients that are undergoing to need all these tests? So we already know that advanced maternal age and repeated implantation failure are enough indications to go for a PGT-A. What are the other things? Now, it's a standard practice to do conventional peripheral blood karyotyping on patients who are facing recurrent pregnancy loss or recurrent implantation failure. What conventional karyotyping does is it gives you information on aneuploidies, on translocations and inversions. So all these things can be addressed with either PGT-A for aneuploidy or PGT-SR for translocations. Then, what about patients who are coming to you and saying that they have a family history of some genetic disorder or they have been diagnosed with as carrier status for thalassemia? So these kind of things are performed by what we call carrier screening. This usually detects mutations, and it helps us ascertain if the mutation is pathogenic or non-pathogenic. It is going to help us ascertain if it is an autosomal recessive, autosomal dominant, X-linked or Y-linked disorder. Now, based on this, we will be able to counsel the patient on their risk factor of developing an affected fetus and giving them 2 options. Either they can go for a donor gamete or they can choose pre-implantation genetic testing for mutations. How does the process work? So basically, we do standard assisted reproductive technology. We do intracytoplasmic sperm injection, where a single sperm is injected into the oocyte. This oocyte is cultured for 5 days in the laboratory. And on the 5th day, a few cells, that is 6 to 8 cells from the trophectoderm, are biopsied using a specialized needle, and a really skilled embryologist is the one who does this. Once these 6 to 8 cells are extracted, they are transferred into a vial, PCR tube, and the embryo is frozen. This PCR tube is shipped at below -20 to -80 degrees temperature to the genetic testing lab. Once a sample arrives at the genetic testing lab, the DNA is extracted directly from the tube because it is not possible to take that minute 6 to 8 cells out of the tube. So it is directly extracted in that particular tube, and then the whole genome is amplified. Sequencing is done on next generation sequencing, which is followed by strong data analytics software, which is AI included, and which is followed by the reporting. So this is an example of a PGT-A report, and what we see on the top figure is a euploid embryo. That means all the chromosomes are in normal content. So if you can see, look at the y-axis, you can see that there are numbers on there. So all the chromosomes are aligned at the baseline of 2. That is, they are in diploid content, which is a normal content. If there are any anomalies, you either find the alignment of chromosomes above or below the baseline. So when it goes above the baseline, as in the next figure, which is of an aneuploid embryo, we can see that chromosome 9 has the chromosomes aligned at the level of 1. That means it's a monosomy 9. And if you look at chromosome 16, it is aligned, I think it's clearer now, it is aligned at 3. So that is trisomy 16. So this is how HGS data is read. The reports usually contain these figures with a complete description of what kind of aneuploidies we are looking at. So when it comes to decision making post PGT, we get 3 different types of embryos. These are the 3 different possibilities. Either we get a euploid embryo. This means the embryo is genetically normal and it is something that we would decide to transfer based on the quality and other factors. And if we find an aneuploid embryo, we do not recommend it for transfer because the chances of it getting miscarried are very, very high. The third category is mosaic embryos. This is considered for transfer only when there are no other options left after due diligence and clear counseling to the patient. And I will get to a little bit more in detail about mosaic embryos in the next slide. So along with PGTA, there is another parameter that is tested, which is a standard protocol and helps us decide which embryo to transfer first. For example, I have 3 euploid embryos and how do I decide which one to transfer? Because all are good morphology and having euploid content. So what we additionally provide is mitoscore. Mitoscore is nothing but the ratio between the mitochondrial DNA and the genomic DNA. Basically, it's a well-established fact in embryos that embryos are supposed to be metabolically silent. So quieter the metabolism, less stress on the embryo. If there are any genetic anomalies, it is known to enhance the activity of mitochondria, basically because you need more energy to fight it. So this was a landmark study which clearly stated that aneuploid embryos had higher mitochondrial DNA content and embryos that did not result in implantation also had a higher mitoscore. So lower the mitoscore, the better. So coming to our problem with mosaicism. Now mosaicism is usually the gray area of PGT, where false negative and false positive reports arise. And this was mainly brought to light by a very, in a couple of years, there were a lot of papers that came out that saying mosaic embryos are resulting in live pregnancies and aneuploid embryos detected by PGT are giving healthy pregnancies. So the question was why and how. So research showed that when we biopsy the trophectoderm, we can't really see which part of the trophectoderm we are biopsying on genetic terms. So it's a blind biopsy of the best looking cells away from the ICM. But in reality, you have, in the left figure, you can see you have 3 possible combinations. Either you are biopsying the complete aneuploid cells or completely normal cells, or a mix of both. And then the original embryo is depicting something else entirely. And there is no way to know it because we cannot biopsy the ICM. Next, what we could see is on the right side, you can see the different types of mosaicism that we can find. So one type is when you have normal ICM and normal trophectoderm. That's the standard normal euploid embryo. Or you get a completely aneuploid embryo. Though that is all black and white. So the gray area falls in a completely mosaic embryo where you can either get ICM mosaic and trophectoderm also mosaic, or else your trophectoderm is normal, your ICM is mosaic, or else your ICM is normal, trophectoderm is mosaic, and your trophectoderm is mosaic, ICM is abnormal. So all these different combinations occur. What we are really concerned about is 2 factors. One, when we give a normal report and your ICM is aneuploid, which can result in miscarriage. So that's why there's a cutoff of about 30%. So embryos are reported to be mosaic if the specimen contains more than 30%, but less than 75% aneuploid cells. Anything outside this range will clearly come as aneuploid or euploid. This is a bit of our internal data that we have from the past year. It is still in its nascent stages and we're still recategorizing it on multiple levels. But this is the preliminary idea of how many euploid and aneuploid embryos we got from our PGTA cycles. Here the patient age was between 30 to 40 years. And what we found was 52% of the embryos were euploid. And a whooping 42% were aneuploid, and about 6% of embryos were called in for mosaicism. Of the aneuploid embryos, we found that 40% had a single chromosome involved, and about 18% had 2 chromosomes involved, 5 to 9% 3 to 4 chromosomes involved, and 28% had multiple chromosome anomalies, which were clearly going to result in miscarriages. Coming to structural rearrangements, that is PGT-SR. Now, chromosomal rearrangements are changes in a normal size and arrangement of chromosomes. That is a structural variation. So this can be found in cases of balanced translocations, Robertsonian translocations, etc. So what happens here is you have in the figure 2 chromosomes, chromosome A and B, part of the chromosome detach and they flip onto each other. So you have part of chromosome B sitting on chromosome A. Now, if this happens without changes in the chromosome structure and the length, it's called a balanced translocation. But if these kind of switchovers cause changes in the size of the chromosome, then it's called an unbalanced translocation, which is not compatible with life. It usually ends up in miscarriage. To understand this scenario better, this is a case scenario. We had a mother who was a carrier of chromosome 3 and chromosome 10, balanced translocation. Her husband was normal karyotype. So there are 4 distinct possibilities of oocytes that she can produce genetically. When cross-matched with the normal component of her husband, she was at a risk of producing 50% of embryos which had unbalanced nature. They either had a deletion in chromosome 3 or duplication in chromosome 3 or 10. Only 25% of her embryos would be categorized as karyotypically normal, and another 25% would carry balanced translocations like the mother, where it would not adversely affect the pregnancy. So overall, this patient had a 50% risk of miscarriage if she would try normally. Now, when we say 50%, doesn't mean that this pregnancy is normal and the next pregnancy will be abnormal. It is that in every cycle when she's conceiving, she is at a 50% risk, which is very high in medical science. So this is a sample report of another case where we had a patient who was a carrier for a translocation between chromosome 2 and 3. We got 4 embryos from this patient, and surprisingly, not surprisingly, we got 3 embryos that were having segmental gain or loss in either chromosome 2 and 3, and only a single embryo that was euploid. So in the image, in the karyo map, you can directly see that there's a segmental loss. Only a small part of chromosome 2 has gone below the baseline. There is segmental loss in chromosome 2, and a small part of chromosome 3 has gone above the baseline. That is a segmental gain in chromosome 3. Next, coming to PGT-M. So this is mainly pre-implantation genetic testing for monogenic disorders, formerly known as PGD. It is addressed to look mainly into mutations and some deletion duplication syndromes. We mainly address, there are about 200 disorders that we provide PGT-M for, and new disorders are usually customized based on the mutation that we find. What we usually get in the common population are inquiries for Duchenne muscular dystrophy, spinal muscular atrophy, beta thalassemia, Fragile X, cystic fibrosis, Charcot-Marie-Tooth syndrome, retinitis pigmentosum, some blindness cases, etc. So what's important with PGT-M is to remember that it is done in conjunction with PGT-A. You cannot do one without the other. You cannot transfer a mutation-free embryo which probably has a chromosome anomaly. It doesn't matter if these patients are infertile or not. So we still have to consider, because even human reproduction itself is not 100% foolproof. So always we need to rule out both these things. And the most important part of PGD or PGT-M is that it's a pre-planned thing. It's not something that you can decide once the embryos are already ready. It needs due diligence and good amount of counseling about the risks and advantages of this procedure. So genetic counseling forms an important part of this process. It's important to counsel patients and get a detailed family history as to what is happening. If it's a clear-cut disorder we are looking for, like thalassemia, it is very clear, and there is a HP electrophoresis report which can tell us the carrier status. Then we can do a targeted carrier screening and find out what's wrong in these cases. But when the patients come with a vague family history and atypical phenotypes, it's quite difficult to ascertain what's wrong with them. So for this purpose, we do carrier screening, that is usually by clinical exome sequencing, or we can do mitochondrial genome sequencing, or we can do whole exome sequencing. That will be the sole call of the clinical geneticist and the genetic counselor based on the extensive family history. Once the clinical carrier screening has been completed, you will find data that is categorized into benign, pathogenic, likely pathogenic, or VUS. So likely pathogenic and pathogenic mutations are actionable by PGTM. It's a clear-cut process that we can proceed with. Once again, VUS happens to be a gray area. VUS are known as variants of unknown significance. This happens when a proper phenotype to genotype correlation has not been established, maybe due to insufficient history, or could be a new mutation that has not been reported earlier. So these things have finally falls to the geneticist to take a call whether it is really something that will affect the patient. We have to take penetrance also, gene penetrance also into account before proceeding with all these things. We also provide the option of going in for gamete donors, in which case gamete donors also need to be screened by sequencing platforms. We need to make sure that they are not carrying any random mutation which can again affect the fetus. Once that is established, we can proceed with the ART cycle followed by embryo biopsy. Once the PGT embryo biopsy arrives at the laboratory, whole genome amplification of the DNA is performed with PCR amplification of libraries targeted at the gene of interest. This is followed by several processes of amplification and enrichment, followed by a targeted sequencing of the gene of interest, followed by linkage analysis. Now, linkage analysis helps us establish the etiology and the lineage from which the mutation is being inherited from the parents. So, any samples from an affected case and the couple and their parents, that is the grandparents of the fetus to be, would prove extremely beneficial and helps us enhance the PGTM diagnosis. If the parents are not available, it is still okay. We can still proceed with the parents and the affected child. That is why DNA storage of patients that are affected with genetic disorders is extremely important, and it proves very beneficial for future diagnostic purposes. So here I have a case to discuss with you, which we performed PGTM for thalassemia. This was a non-consanguous couple who had earlier done HPLC electrophoresis for thalassemia. They both were known carriers of thalassemia minor, and they came to us for thorough genetic counseling was done, and targeted carrier screening for HBB gene was performed. So we found that the husband was a carrier of a 619 base pair deletion, and the wife was a carrier for a G2T mutation. This was followed by IVF and biopsy. We actually got 5 embryos from this patient. The first embryo biopsy did not yield sufficient amount of DNA, so that was excluded. We had the second embryo which showed multiple chromosome abnormalities. Now this is not an infertile couple, okay, and they're in their early 30s. Still, there was one chromosome with multiple chromosome anomalies that we could not use. The other 3 embryos showed normal chromosome complement, but E3 and E4 were carrying the mother's mutation, and E5 was carrying the father's mutation. So they decided to counsel the patient, and the patient decided they wanted to transfer an embryo with the mother's mutation. So I have 2 embryos with the mother's mutation. So which one do I select? This decision was based on the mito score. So the mito score of E3 was lesser than that of E4. So E3 was selected for transfer. Unfortunately, it did implant, and the patient had a healthy baby who is now a carrier for the mother's mutation and is not adversely affected with thalassemia major. Moving on to the next type of PGT, that is the new technology that is available now. It's just coming into the market and making a lot of noise all over the world, is a non-invasive PGT, abbreviated as niPGT, and it is focused at using the culture medium. So the embryo is in a constant equilibrium with its environment. It's constantly taking up things and releasing things into it. Things by, I mean, not just nutrients. It is releasing microRNAs, it's releasing proteins, it is releasing DNA. mostly cell-free DNA. So this is what we are testing when it comes to non-invasive PGS. So usually the culture medium that is used for it is post day 3, mainly because day 3 embryos are known to carry higher incidence of mosaicism. That is why the culture medium is changed on day 3, which is a standard practice in IVF. So we can use either day 3 to day 5 medium or day 4 to day 5 medium. Or if I have a blastocyst that is already frozen earlier, I can also use a day 5 to day 6 medium. So the culture, it doesn't matter on the duration of which we use a culture medium. Basically, the longer the duration, the better. If you are an IVF center that is doing a one-step culture, that is from day 1 to day 5 in a single culture medium. If it's a single embryo culture, we can take that also. That is not at all an issue. But it is mandatory to culture the embryos in single droplets. So on the day of sampling, we can remove the embryo, freeze it, and then extract the culture medium and send it to the laboratory for testing. Another important thing we need to consider is maternal cell contamination in such cases from cumulus cells. They need to be washed away. And IVF embryos are usually not preferred because it means that you have to wash the embryo multiple times. And if any of the embryologists are on this forum, they would know that we wouldn't want to disturb the embryo that many times. So ICSI embryos usually have minimal such issues. And by day 3, all the cumulus cells are shed. So it's an ideal sample. The difference between the standard biopsy and the culture medium is the amount of DNA is very less. So the amplification of this DNA was achieved by a groundbreaking technology called MALBAC, that is multiple annealing and loop-based amplification. So what this technology does is it uses degenerate primers, and after every cycle of amplification, the product that is the first cycle of amplification itself, the product that is generated forms an amplicon. It forms a hairpin loop, and again the next cycle of amplification uses the main DNA. So it's a quasi-linear amplification which helps to overcome errors during whole genome amplification. The evidence for this technology was very strong. There were, in the last couple of years, there were excellent papers that came in publications like PNAS that showed that this technology addresses the embryo at a whole. So it helps to overcome mosaicism by a lot. And it also, there were a lot of live births also reported. China has been doing this since 2015. They run about 30,000 embryos on non-invasive PGS. European countries are also doing this. And even in the US, approximately 5,000 to 8,000 of embryos are being screened per month right now. And this technology is going to be available in Asia in the next 6 months or so, once the pandemic is over and everything falls into place. So this is something that all of us are looking forward to. The most interesting part of the studies that watched for this technology is that it showed 100% sensitivity and specificity. And there was a comparison that was done with the culture medium, the trophectoderm biopsy, and the whole embryo. So the concordance of the non-invasive PGT with the whole embryo was as high as 93%. It reduced false positive rates from standard PGT to 20%, and there were no false negative rates. Another slight variant of minimally invasive PGT is where you put a needle into the blastocele cavity, like I said before. The blastocele fluid is also extracted, and this showed about 90% concordance to the whole embryo. So this was another alternate to the culture medium also. These all can be done without the use of a laser. And the most recent advance that is in process of being commercialized is PGT for polygenic disorders. It is labeled PGT-P or expanded PGT. This group published a very beautiful paper for proof of concept in fertility sterility in 2018, and 2019 paper showed that they have optimized the concurrent PGT for monogenic disorders, structural rearrangement, aneuploidy, and polygenic disorders in the same embryo biopsy on the same platform at the same time. So the polygenic disorder list is mainly addressed for conditions like multifactorial conditions like CVD, diabetes, hypothyroidism, and they've included a bunch of cancers. So this is something that is still in development, and it will take some time for it to be accepted, mainly because as such, once pre-implantation genetic testing is done on embryos, we have very few embryos to select from, and then this further reduces options. So it will take some time. One small thing I would like to emphasize in the middle of all this is genetic counseling. Geneticists play a key role in PGT cycles, no matter what kind of PGT you're going in for. So they will be able to explain to the patient in detail the risks and advantages of PGT. So there's a risk of a failure to generate report due to both biopsy quality and the embryo quality, and the risk of embryos not being available for transfer. What if we find all aneuploid embryos? That is a possibility. We can never deny that. The freeze-thaw survival, though vitrification is extremely standardized, we have got very good success rates. It is quite possible that one or two embryos do not survive freeze-thaw. And also the limitations of diagnostic procedure. For example, if you're doing PGT-A, we cannot look at triploidy. That cannot be found, because if all chromosomes are on 3 copies, NGS cannot detect it. Okay, and if we find polymorphic segments on the karyotype after the baby is born or in prenatal testing, it cannot be detected on NGS. Or if there are deletions and duplications less than 10 MB, even that cannot be done on PGT-A. And also what's important is patients need to be counseled that prenatal testing is an important part of PGT, mainly because we should understand that the technology is advanced so much that we are able to give complex genetic diagnosis, what we would usually do on peripheral blood, by just observing 5 to 6 cells in an embryo. So there are some limitations, and there is a risk of 5 to 6% of something getting missed out in all these things. So it's better that a prenatal diagnosis be performed for such cases. Just to emphasize how technologies are different from each other, we have conventional karyotyping, which is done by G banding. It has a resolution of 5 to 10 MB. It can pick up translocations, balanced, unbalanced, inversions, polymorphisms, and deletion duplications above 50 MB, mosaicism at 10%. But this can be done only in peripheral blood. It cannot be done on embryos. So FISH is an outdated technique, so no point in discussing that now. Right now, we have SNP arrays and NGS. NGS is the king of the processing platforms right now, mainly because it's able to do segmental aneuploidies that are arising out of unbalanced translocations. It can help study aneuploidies and mutations on the same platform. It can pick mosaicism as low as 25% also, and has very low false positives. So to conclude, application of PGT is rapidly gaining popularity, and people are beginning to accept it because in indicated cases, it is showing good progress. It is helping to a lot of patients with advanced maternal age. It is reducing the time to pregnancy. It is improving pregnancy rate by almost 30% in some centers. It is reducing the risks of miscarriage, and also PGT-SR is something that is really beneficial for patients with translocations, because by the time we get patients who are translocation carriers, they would have undergone 4 or 5 miscarriages, and it's a painful process. And PGT-M is ideal for patients who are carriers of monogenic disorders. Thank you so much for your patient hearing.
Thank you very much, Dr. D'Souza, for your excellent presentation on advances in the pre-implantation genetic diagnosis. I think now the audience is open for the questions and answers and some comments or discussions. So please have your comments, questions. So Dr. D'Souza is hoping to answer.
Swaminathan sir is asking the percentage of mosaicism and its implication in the results observed. So we find as our internal data shows, we find about 6% embryos that are mosaic, and reportability of embryos that are mosaic is usually considered above 35 to 40%. With mosaic embryos, we never know if they will give us a pregnancy or not. So patients need to be counseled that when we have no other option left, we are going to go for mosaic embryo transfers, because we do not want to waste embryos. They're precious commodities. And we need to tell the patient that there is a risk of miscarriage, and prenatal testing is mandatory in such cases. If an informed decision is taken along with the patient, yes, we can transfer mosaic embryos and pray and hope that it has a good quality ICM without any aneuploid cells, and it can result in a successful pregnancy. This is mainly because embryos have a capacity to expel abnormal cells into the trophectoderm. That is the main reason we have trophectoderm ICM mosaicism. Somebody's asking on the sensitivity and specificity of niPGT. So the specificity of niPGT is 100%. The sensitivity is around 98%, which is acceptable. Our main concern with using niPGT is the concordance rate. How is it actually depicting the embryo? So niPGT matches trophectoderm biopsies at about 85% concordance, and it matches the whole embryo content at about 93%. So in centers where there are no lasers and you don't have a skilled embryologist, and right now, like we are in a lockdown, we are not able to get an embryologist from another city.
Am I talking about embryo gradings?
Sorry, sir. Hello. Yeah, so in such cases, non-invasive PGT can be used.
Hi, Dr. Fiona. Good
Yes.
evening. Good afternoon. Good afternoon. I'm one of the clinical geneticists in Sri Lanka. So just want your opinion. Thank you very much for your excellent presentation. I just wanted to know your opinion regarding the patients with polymorphic variants. What is the rationale? How it is ethical for us to offer them PGS? Like, no, there are so many couples, they present with kind of like an unexplained subfertility, and
Yes.
maybe fail with a few cycles of IVF. So do you justify to offer them PGS?
Actually, ma'am, I would like to emphasize about polymorphic variants, because we find this a lot of cases come to us with polymorphic variants, and polymorphic variants are present in normal population as well as the infertile population. However, a lot of RPL recurrent pregnancy loss cases we have some indication for polymorphic variants, but NGS cannot detect polymorphic variants. That part of the genome will not be mapped, and it is not really necessary to offer them PGTA because of the polymorphic variants. You can offer them as an age factor, or if they're an IVF patient, they might get an aneuploidy, whole chromosome or segmental, then you can offer them. But if it's a polymorphic variant, really the things that we can do are quite limited. All we can try is the standard testing, or if there is no other way and they are running out of time with the age factor, we would also suggest an option of donor gametes. Any other questions?
So if you have any more questions, please you can put it into the chat, or you can just on your mic, and you can ask the questions. So we have a little bit more time. Okay, it seems that there are no more questions and other comments. So I would like to take this opportunity to thank Dr. Fiona. So thank you very much, Dr. Fiona, for your excellent presentation. I hope everyone had informative and timely update on current trends in pre-implantation genetic diagnosis.
Thank
So
you,
please,
sir.
please notice that the recorded version is available in the Human Genetic Society website. That is humangeneticsociety.lk. So it is also in the chat. You can save the chat and you can directly go into the website. Then you can download the webinar from there. Yeah, when we will upload it. So. So I would like to take this opportunity to thank our speaker, Dr. Fiona, and thank you very much, Fiona, for your accepting the invitation for today's webinar. I hope this would give a timely update on the pre-implantation genetic diagnosis. Actually, it was very informative and with the available new trends and practices.
If any of you have any additional questions, you can send me an email or you can email Dr. Lahiro on the Human Genetics Association email, and we would be happy to respond.
Definitely. I think we will upload the, so you can log into the Human Genetics Society.lk website, and even you can send emails to the Human Genetics Society.lk email, and we will happy to answer all the comments with the help of the Dr. Fiona. And at the same time, I would like to give my gratitude to the President and the Executive Committee of the Human Genetics Society for arranging this valuable series of academic webinars. And also, I would like to give my acknowledge to the Dr. Chamika and Dr. Roshan Hevapatirana of the Postgraduate Institute of Medicine in Sri Lanka for their support and arranging the the webinar. And also, finally, I would like to thank all the participants. So I think it is a greatly attended webinar, and I'm happy that our monthly webinars, the coming month, and also the after that, so you all can join and have these important updates on the current trends. Okay. Thank you very much. Thank you, Fiona, and thank you all.
Thank you.
That better hatched and hatching.
Sorry, sir. Can you repeat the question, please?
What is the best time of embryo and PGT?
Okay.
That hatch and hatching and other.
So usually I think the question is on embryo biopsy timing. Is that right?
Yes.
So we would prefer giving a laser shot the previous day night or early morning on day 5.
Okay.
So that a small breach is made in the zona, which helps the embryo herniate out. So that is a hatching embryo. Once a few cells have herniated out, embryologists usually keep an eye on it. And biopsying these embryos becomes much more easier. It is less invasive and less stressful. Nonetheless, skilled embryologists can directly go in for biopsy of expanded blastocysts. And completely hatched embryos are slightly difficult to handle because of the absence of zona. That is why a timely biopsy is very crucial in making sure that the embryo incurs minimum amount of stress during the biopsy procedure.
Thank you. Can another question?
Yes, sir.
Hello.
Yes. Hello.
Hello.
Yes, please tell us.
Now, what is the best time of embryo PGT and freezing?
Freezing.
Yes, yes.
Actually, embryos can be transferred for freezing immediately after biopsy, or you can
Yes.
wait for, if you are more comfortable with expanded embryos. Usually embryos tend to collapse during the biopsy procedure. This is normal. It does not cause any kind of dangers to the embryo. So you can wait a couple of hours, 1 or 2 hours, to wait till the embryo expands fully and also biopsy, also freeze. But collapsed embryos are very good at absorbing the cryoprotectant, so freezing them becomes easier. So you can freeze immediately after biopsy as well.
Can I thawing for frozen embryo transfer? What is the time?
Usually, embryo transfers are done approximately 2 hours post thawing. Minimum 1 to 2 hours we can wait post thawing for embryo transfer. Another thing I would like to add in is when it comes to biopsying the embryo, it is always advised that the person who is going to biopsy the embryo has at least practiced on 50 embryos, and the embryologist should be trained skill-wise to ascertain embryo survivability. So when you are practicing embryo biopsy on leftover embryos that you have, you need to make sure that these embryos survive your biopsy and they have to survive freeze thaw. Only when about 50 of your embryos have survived this, then you know that at least you are at a success of 90%. And tubing of a biopsy sample is also very important, because we go through all this trouble, and if you're not able to tube it properly, it becomes the whole purpose of the process is redundant. And you need a lot of practice when it comes to tubing, because it is handling very small biopsy sample. And an ideal biopsy sample will have a cluster of cells, not individual cells that are separate. So when there's a cluster of cells, the chances of us giving a successful report are always higher.
Hello?
Yes?
How many can an embryo, frozen embryo transfer? It will be PGTA?
Usually PGTA advises a single embryo transfer.
Okay.
So you can generally, you don't, when you've gone through all this trouble to genetically test the embryo, the whole purpose was to get a genetically normal embryo and to avoid multiple pregnancies. So usually PGTA is followed by single embryo transfer.
It depends of women age for embryo transfer.
Sorry?
It depends of frozen embryo transfer for women age.
No, age is not a factor when it comes to FET. Age is a factor when it comes to the quality of embryos you get, because as age advances, the disjunction between chromosomes are higher. So the oocytes that are produced by patients as they progress in age have higher antibodies.
Ma'am, can I question another question?
Yes, please.
Okay.
Hello? So there is somebody asking, do genetic guidelines are well established? Yes, genetic guidelines for reporting PGTA and PGTM are quite established, and they're very clear, because we are taking a call on life before it begins, and it's a matter which is taken very, very seriously by everybody. So we have clear guidelines that state, even mosaic reporting has guidelines. So most of us in India, we follow European guidelines, and European Human Genetic Society for such guidelines. So the bioinformatics softwares play a very critical role in all of this. So these are AI-enabled self-learning softwares. So the more data you feed into them, the more sensitive they become.
Okay, thank you very much, Fiona. So there
Yes.
are no any more questions now, but I hope that if you have any questions, please email into the humangeneticsociety.lk. And also, I would like to request from you to share your feedback and your comments about this academic webinar into the humangeneticsociety.lk. Okay, thank you very much, and
Thank you.
good day for everyone.
Thank you.